Okay, good afternoon, everybody. I was in a meeting until literally 30 seconds ago. So let's see how we go. Uh, I'm going to tune in now to Lockdown Bioinformatics. Okay, so we're day 35. And for those of you that don't know, don't follow me on Twitter for some inexplicable reason, we do now have a group Slack workspace for lockdown learning. I posted an invite link on Twitter this morning, and I've seen quite a lot of people have joined, but you're all more than welcome. It would be good to see lots of people in there. I think it would be there's lots of interesting things that possibly opens up. So it's I would like it to be a place for where support can happen that isn't necessarily my Twitter DMs or the live chat, which tends to terminate quite quickly after the stream is finished, whether I like it or not. So it's a bit more persistent in terms of the conversations that can happen. And also, hopefully for those people who aren't following live, it gives them a forum where they can ask questions sort of as they occur. And a little bit of a forum potentially for peer support and things like that, as well as the kind of top-down support that we've been doing so far. So do join in there if you so desire. Obviously, the live chat on YouTube. Okay, and so to today, to day 35, and where we're at is we spent yesterday producing these reasonably standard plots for RNA-Seq data. So we looked at the MA plot, which you can produce using DE-seq's plot MA function fairly easily, and then we looked at a more complicated to produce volcano plot, which has been around volcano plots have been around since the days of microarrays and presumably beyond. But they tend to be quite nice ways of highlighting interesting changes in a high throughput experiment like this. So that's the plot that we've got there on the screen, which is the interactive version of our volcano plot that we produced towards the end of yesterday's lesson. And we used the Plotly library to add interactivity to our plot, so to make it possible to hover over the points and have information appear. I seem to have managed to zoom into that slightly by accident there. So we get this sort of interactivity available to us using Plotly, which is quite nice. And the other feature of this was our ability to add arbitrary labels to these little hover over tooltips that we get in this interactive version of the plot. So we added in the ensemble gene IDs as an extra little bit of annotation on our plot just before we finished yesterday by adding in our aesthetics, our global aesthetics, in our call to ggplot, the ggplot function, we added just an additional aesthetic which we're not using anywhere in the plot. We're not using it in any of our geoms to inform any of the kind of base visualization, colors and shapes and sizes and things like that. But because it's in the aesthetic, it's available to that tooltip in the interactive version of the plot, and so appears there, which is quite a nice little feature for that Plotly gives us. The limitation of that at the moment is that ensemble gene IDs, while they are very useful as identifiers because They're unambiguous identifiers. They uniquely identify one gene, and they can be used in lots of ways. They're certainly not human readable. I mean, there are genes which I come across, although I analyse data from lots and lots of different groups here in Newcastle, because that's the nature of working in and running a support unit, is you work with lots and lots of researchers. And so I've analysed data in lots of contexts, from lots of organisms, lots of different types of experiment, lots of different conditions. But even despite that, there are genes which you come across over and over and over again. Genes which occur as important genes in all sorts of systems, and we do a lot of work with humans because we're a medical school, so we have a lot of human experiments. So I work with human ensemble gene IDs an awful lot, and we come across the same sorts of genes in lots and lots of experiments. It's amazing how much the inflammatory system is involved in all sorts of diseases, whether they're recognised as inflammatory diseases or not. But despite that, I could not tell you a single mapping between an ensemble gene identifier and a human readable gene name or a description for that gene. They do not fit gene IDs onto a plot like this. Is of limited value. Okay, it gives me an identifier I can look up if I've got a browser handy and I can type the identifier into the browser. But actually, it would be better to systematically have a more human readable identifier for the genes both on our heat map and just more generally, just for working with this data downstream. If we're going to start exporting tables of differentially expressed genes and things like that, we want more annotation available to us in those tables than we currently have, than just the ensemble gene ID that we currently have. And so that's the motivation behind what we're going to do today, which is we're going to take these ensemble gene IDs, and we're going to do it for all of the 15,000 genes in this heat map to show, in this volcano plot, to show that that's possible. We're going to take the ensemble gene IDs and we're going to map those ensemble gene IDs to other annotation. So features of those ensemble gene IDs which might be useful to us. Things like gene symbols, gene descriptions, maybe chromosomal locations for genes, and some other things besides. So that's our goal for today, is to map our the ensemble gene IDs that we have, which are the only identifiers for our genes, to other information to make our tables richer and our plots richer, and so on and so forth. Okay, so how are we going to get that annotation? There's lots and lots of ways of doing this. There are databases that are distributed, databases of ID mappings that are distributed by Bioconductor that you could use to essentially do a lookup. There are packages which are distributed which contain the annotation tables for particular releases of ensemble. So there's a chap who I can't remember where in the States he works. He's called Stephen Turner, and he does a job very similar to mine. I think he runs a core facility, and he has packages on GitHub which are basically annotation tables for various releases of ensemble and of GenCode. But we're going to use a package called BioMart to do this. And BioMart is a web service. It's an online service which does ID mapping and lots of other things besides. And it's an add-on to Ensemble. It's a branch of Ensemble. So I'm just going to pop the Ensemble website up to show you. So this is the Ensemble website, ensemble.org. And we've not really talked about genome browsers or anything like that today. We may think about that in a future lesson. But Ensembl provide a genome browser, so you can go into Ensembl and look at a particular gene and find its genomic location and see what other genomic features are co-located with your gene of interest, and lots of other things besides. But they also distribute annotation, so you could download gene sets from Ensembl if you wanted to. We downloaded the GenCode gene set, which is actually very closely related to the Ensembl gene set. They're very similar, but GenCode to me, do a slightly more regimented release of kind of reference annotation, and it's easier to find than the annotation.ensembl release. But the other thing that they provide is this BioMart service, and there's a link to it in the toolbar at the top here. So there's a link to BioMart there, and BioMart is actually quite a wide-ranging web service, but essentially it's a way of interconverting between different data sets. So you can use BioMart via the BioMart web data set. You can pick, you can add filters, so you can say, I want data for a certain set of genes. So you can plug in filters and say, well, I want this particular list of genes. You can paste a list of genes into here and retrieve the annotation for those. And then you can, and there's lots of different types of filter that you can apply. You can put in all sorts of things in here and filter by lots of different features, lots of different types of feature, and so on. BioMart's quite useful for that sort of thing from time to time. And then for everything which passes your filter, whatever your filter is that you've put in, so maybe I've put a list of Ensembl gene IDs in here. In fact, let's just grab 3 or 4 Ensembl gene IDs, and I can show you. So let's have a look at row names. Filter. Oh no, we changed it with df1, didn't we? Filter df1 dollars ends gene. So that gives me that column as a vector. Let's just get the first 6 Ensembl gene IDs from that list, from that vector. I'm just going to copy that and paste it into this box. I'll just have to tidy it up, because obviously cutting it out of R and pasting it in here is not, it doesn't really give me enough format that's terribly useful. So I'm just tidying up so it's just a single Ensembl gene ID per line. So there we go. So that's the first 6 Ensembl gene IDs from our results, from our filtered results data frame. So we've pasted those in there, and then we can retrieve from the genes which match these filters, we can retrieve certain attributes. Okay, so we can, at the moment, we're getting these various attributes for this list of 6 genes. So we're going to get the gene stable ID, so we're getting back the identifier that we've plugged in. We're also getting the gene ID stable version, the transcript stable ID, so any transcript IDs which match those gene IDs, and then the transcript stable ID version as well. So you can see immediately we could probably use this for making our TX to gene data frame that we needed for the input for TX import. But there's lots and lots of attributes that are available to you as well. So we don't just have to get those 4 things that were suggested by default. We could get lots of other things besides. So we can get gene name, for instance, or we could get, you can see there's gene ontology terms in here, reactome IDs, KEGG pathway IDs. There's all sorts of things in here that might be useful to us. For now, I'll just add gene name to this. And then once you've decided on your filters and your filters that you want to apply and the attributes that you want to retrieve, then you can click on results, and it will give you, first of all, it gives you a little preview of your results table, and then it also lets you download that table, so you can export that table out to a TSV file or a CSV file or anything else. You'll notice that for each gene ID that we uploaded, we've got a number of results, and that is because there are many transcript IDs for any one given gene ID. That's mostly the reason why we get lots of results in this table. So we've not just got 6 rows in our results table, we have lots of rows because each gene is generating many results, because each gene has many transcripts and may also have many gene names as well. But we've got gene names here which are more human readable. Okay, so that's the idea behind the BioMart web service. And you've got this browser tool, which is actually very useful. But the BioMart API is slightly more complicated than the UniProt API, which we used before to download UniProt data. This is much more of it is a programming interface. It is a way in which you can define these queries within a program and retrieve the data in your code. So we could write a Perl script or a Python script that uses that API and retrieves the data. But because we're working in R, we're going to do this in R, and handily somebody has already written a package which maps this web service into convenient function calls in R for us to retrieve exactly this kind of data in our R session, in our R script. Okay, so in order to do we need to be able to install the BioMart package. It's a Bioconductor package, but because we're using Bioc Manager, we don't even really need to know that. I'm just going to close my viewer interface because it slows my R session down considerably. So I'm just going to clear that so that I can work at normal speed, basically. So we need to install the BioMart package. We don't need to worry about where it comes from because we've got our Bioc Manager install function for installing packages wherever they come from. BioMart is capitalized like this, so it's all lowercase apart from the R, which is uppercase, and that's because we're using BioMart from within R, so the R is capitalized. Or at least I think that's what was in the package developers' minds when they named it. So BioMart is all lowercase apart from capital R, and the capitalization is important. So this is going to install the BioMart package for me in my installation of R. I still don't want to update these packages, so I'm going to say no. So then I need to make the package available to me, and if you remember, I am whenever I load a package into my current script, I always do so at the top of the script, and that's so whenever I come to this script again in the future, I can see which packages it requires, and that list of packages is getting quite long now. Now, quite a few of those are tidyverse packages, and we have actually loaded nearly the entire tidyverse now. So the base tidyverse is 6 packages. We've loaded 5 of them, I think. So we've loaded Reader, dplyr, Magritte, ggplot2, and Tibble. So we're only 1 package away from loading the tidyverse. So now might be a good time to tidy that up. So let's do that. That's library tidyverse. And let's just get rid of the tidyverse packages individually. So that's Tibble, ggplot2, Magritte, dplyr, and reader, and that makes things considerably neater. And then the package that we just installed is BioMart with a capital R. Okay, so that's our new package that we've just installed. What is that? There is no package called AskPass. That's interesting. Should have taken care of all of the dependencies for us, so it's slightly odd that when trying to load it, it's failing to load a dependency. Well, let's try installing that. Whoops. Like that. See what that achieves. No. Try again. So that worked that time. I don't know why that dependency wasn't resolved by install. It really should have been. So that's an interesting one. But anyway, that BioMart is now loaded for me. So we've loaded BioMart, and now we need to know how to use it. So BioMart is quite complicated. It's got several steps for us to get to the data, and those steps are quite well described in BioMart's vignette, in BioMart's documentation. So let's have a look at that. Let's do $BioMart. Let's just search for the package. And so here at the top of that, we get the vignette. So the vignette is like the user's guide. So that is our kind of guide to using this package. And I would say that for an awful lot of packages, especially in Bioconductor, they have very good vignettes, which are very descriptive of how you would go about using that package. So the BioMart vignette we'll have a look at in a second. I have never shown you the equivalent for DSeq2, and I really should have done, because again, let's just do $DSeq2 and stop its worst autocomplete. DSeq2, the vignette for DSeq2, if we open that, is we will. Okay, what I'm going to do is I'm going to open this in a new window. So that opens the help interface in its own little window. This vignette is a substantial document with lots and lots of information in it as to how you work with DSeq2 and how you set up all sorts of different ways of comparing data. There'll be all sorts of things in here, and there's lots and lots of technical information. It's a very useful document, and it is generally true. So there's an MA plot. It is generally true that vignettes for Bioconductor packages are really decent. Documents. So there's plot counts, which we've looked at. There's an equivalent ggplot plot. The DSeq one is particularly good, and has lots of information in it. So we got some heat maps and things like that here, which we'll come to later. So it's worth browsing. The BioMart ones I think is okay as well. So we'll go back to the BioMart one now. But if in doubt, if you don't know where to start with a package, if you know that there's a package that exists which is designed for analysing the kind of data that you want to analyse, then have a browse of the vignette first. It's always a really good place to start. So let's have a look at the vignette for BioMart. Again, I'm going to pop this out in a new window just so I can put it up full screen. So the kind of mode of operation is covered in sections 2 and 3. Section 2 is what we really care about right now, which is how to connect to the appropriate database and dataset. You can see all of these different functions. So the function calls are described, and some example results and things like that are all here. And so we can, if we want, work through this this vignette to figure out how to get from A to B to C to get us to a point where we can query BioMart. So what I'm gonna do is I'm gonna take this off the screen, but I am basically going to work through this document because I think it's reasonably illustrative. So we've loaded which marts are we able to load. So the function for that is list marts. And it lists, so at the moment, the current version of Ensemble is Ensemble version 100. There's Ensemble datasets available for human, the Ensemble genes dataset for mouse strains, for variation and for regulation. We want the Ensemble genes BioMart. And it doesn't quite tell you how it gets from A to B here, but the way to use that mart is to run ensemble. So this is a variable that we're going to create called ensemble, and so rather than list marts, now we want to use a mart, and the mart that we want to use is called ensemble. Sorry, that should be a character, so that should be in quotes. It's not obvious to me from the results of list marts here how you get to the mart that you want to use being called ensemble. But that's what's in the vignette, and that's what works. But we do need to do something else here. We need to be specific about the version of ensemble that we want, because as I mentioned just a couple of minutes ago, the current version of Ensemble is version 100, which was released about a week after we started working with our RNA-Seq data. The version of GenCode that we downloaded is GenCode version 33. GenCode version 33 was built off Ensemble version 99. Ensemble version of 100 came with a new version of GenCode, GenCode version 34. But we're working with GenCode 33. And if you get the wrong version of the annotation, you will end up having gene IDs in our dataset which aren't present in Ensemble version 100, because Ensemble has moved on. Ensemble has changed. It may have changed very subtly, but it will have changed between version 99 and version 100. And one of the things that changes is they update gene identifiers. Some gene identifiers fall out of use, and some new gene identifiers come into use. I've never been entirely clear why the flux is quite as quick as it is, but nevertheless, there will be differences between Ensemble 99 and Ensemble 100. If we load the default version at this point, we'll be loading Ensemble 100 as list marts showed us. And we don't want to do that. We want to load Ensemble version 99. So we can do that by giving it a version argument. So version equals 99 will load Ensemble version 99. I'm going to call my my BioMart variable at this point. I'm going to call it Ensemble 99. So this function that we've got here on line 180, this will connect us to BioMart. So it will connect it. This is like when we loaded up the BioMart. So we can make an analogy here to the BioMart website. So if we go back to the front page of BioMart, hold on, bear with me a second. Ensemble org BioMart. So I'm just reloading BioMart. Here we go. Oh, it's taking me back to. Let's go right back out to the top. I can't even get out of here. There was a page before this where we had to pick which BioMart we wanted, and it was the equivalent of picking between these four: the ensemble genes, mouse strains, ensemble variation, and ensemble regulation marts. We picked ensemble genes 100. So there you go. There's the options there. So what we've just done, we've effectively picked an option from this drop-down box. Okay, so we've picked the ensemble genes 100 entry from this drop-down box. The next thing that we have to do is we have to pick the data set that we want from this drop-down box. Okay, so that's the next step that we have to do in R, is the same as the next step that we have to do in the web interface, which is we have to pick a data set. And as you can see, there are an awful lot of data sets. We want the human one, but let's just have a look at how we do that in R. I'm going to drop that back down. So we have a function in BioMart which is list datasets, which takes an argument of the mart that you're currently attached to, and will list you all of the datasets that are available for that mart. I haven't actually run the use mart. Why didn't that work? That worked when I tested it this afternoon. For goodness sake. Let's try BioMart equals ensemble. Okay, there you go. That's better. Oh no, that doesn't work either. This worked this afternoon when I tested it. This is annoying because... oh, I know why. I know why. It's because we don't use usemart. That's my fault for using the vignette and rather than using the thing that I tested this afternoon. If you want to use ensemble, there's a shortcut. And that shortcut is use ensemble rather than use mart. And this will connect to the ensemble BioMart rather than a generic BioMart available on biomart.org or something like that. So use ensemble here will work to connect to the BioMart ensemble instance that we're working with in the website. Okay, so sorry about that. That was me. That was my fault. Okay, so this has connected us to the ensemble BioMart, version 99 of the ensemble BioMart, because that's important. Okay, so now we can list the datasets available in that version of BioMart, and that returns us a data frame actually of a list of all of those data sets. There's 202 of them. If we want to look at that in slightly more depth, we can use view because it's a data frame, so we can look at it like that, and that shows us all of those data sets there. So this is all of the data sets that are available in the ensemble BioMart, and you can see there are lots of them. The one that we want is the human one. So let's see if we can find it in this list. There it is, H. sapiens gene ensemble. That's the data set that we want. So we want to connect to that data set. And to do that, we use the BioMart function. So we're going to recycle our vector name, use dataset. The dataset that we want is that H. sapiens gene ensemble that I just copied out of the table. And the mart that we're using to get it is this connection that we made on line 180. So the mart is ensemble 99. So that's the variable that we've already created on the previous line. Okay, so this will select that data set for us. And that is the equivalent in the website of dropping down this choose dataset and picking human genes. Okay, so that's effectively what we've just done in our R session. And that then pops up in the website, this pops up this thing to populate with filters and attributes. And we can do the same thing in R. So the way that we now get our annotation, and I'm going to do this, what I'm going to do now is I'm going to do the same query in R as we did previously in the web form. So I'm going to get those first 6 ensemble gene IDs. I'm going to get their ensemble gene ID back and the ID version and the transcripts and the transcript version, and I'm also going to get the gene names. Okay, so I'm going to get those attributes out. To see what attributes are available in the data set that you're using, again, BioMart gives us a function and it's list attributes. And the argument for list attributes is, again, your current connected BioMart instance. This will produce a very large table, over 3,000 rows. So again, we can use view to give us a more browsable version of that table. And so these are all of the attributes that are available to us. So we want the first 4, ensemble gene ID, ensemble gene ID version, ensemble transcript ID, and ensemble transcript ID version. And then we want the gene name as well. And let's just find that. The gene name is the name of that column in our attributes is external _ gene _ name. So they are the 4 attributes that we want to retrieve. Okay, and the way that we do this, so in the website we would find those boxes and we would fill them in. The way that we do this in BioMart query is in R is to use the function get BM. Okay, so get BM is the function which applies the filters that you give it and retrieves the attributes that you ask for. So we're going to ask for 5 attributes. We're going to give it a filter in the same way as we gave it a filter in the website as well. So the first thing that we need is the attributes. So this will be a character vector, so constructed with C, of those 5 attributes that we just identified in this table. So we had ensemble gene ID. Okay, that's the first one. Then the second one was ensemble gene ID version. And then it was ensemble transcript ID. And ensemble transcript ID version. And then the last one was, here we go, it was external gene name. Okay, so that's the 5th one that we want. So that's our attributes. They're the things that we want back from BioMart. So then the next thing is filters. So that's what do you want to filter on? What information in BioMart do you want to filter on? Effectively what's the column in the big BioMart table that you want to use to apply filters? And again, these are available. You can find out what filters are available using list filters for your BioMart dataset, which you've currently got attached. So here are the filters. And like the attributes, there are 3,500 attributes available to you in BioMart. You can filter on just 434 different attributes. So, just a mere 434 things that you can use for filtering. We want to filter on ensemble gene ID because the ensemble gene ID that's the identifier that we have available to us for retrieving this information from BioMart. So our filter is ensemble gene ID. I just want to find it in here just to prove that it's available. Here we are, ensemble gene ID, which have a gene ontology identifier. So you require with go to be true, and things like that. There's lots of kind of options available to you for filtering data from BioMart. So again, filters takes a character vector, so C, and this time it's just going to have a single entry in it, ensemble gene ID. So if we think of BioMart as a big table, a big lookup table, it's a bit more complicated than that. But if we think of it like as a big lookup table, what we're doing in the attributes is saying these are the columns we want to retrieve from that table, and the filters is based on this. We want to retrieve that based on this column. So this is the column we want to look up, and then the things that you want to look up in that column are passed to getBM as the values. Okay, so the values are then the things you want to look up. So these are the ensemble gene IDs that we want to look up in our big table of BioMart. And so this is the values is the equivalent in the BioMart web interface of what we pasted into this filters box here. So these 6 identifiers that we pasted into this box, these are our values. So that's what we want to give our call to getBM as the values. So if you remember, I retrieved these in R right at the beginning. So let's just go back through our history a bit. There we are. Those are our values. So that's the first 6 gene IDs from our ends gene column in our filter df1 data frame. So that's our values. And then the final argument that we need to pass to getBM is our BioMart, is the BioMart that we're connected to, which contains this data. So that argument is mart, and the value that we give it is our BioMart, so ensemble 99. Okay, so this will go to the BioMart website. It will do exactly the same query as we've done in the web form here. It will look up those 6 ensemble gene IDs, and it will give me back these 5 attributes that map to those 6 ensemble gene IDs. And it will then give me the equivalent of this table. But obviously in the BioMart web form here, we're looking this up in ensemble 100, whereas in our BioMart lookup in R, we're doing this in ensemble 99. So the results may be subtly different. I hope that they wouldn't be very different, but they may be subtly different. So this, I'm not capturing this as a variable at this point. So this is going to retrieve me a data frame, and it's going to just print it out into my console. So let's make my console take up the full. So they're the 6 that we're looking up. So if we execute that command, there we go. So that is our data frame of the results from BioMart for those 6 ensemble gene IDs. So all of their transcript IDs with the stable version numbers and with the external gene names. So that table there has 34 entries. Our table in the BioMart web interface If you click on count, it tells you how many results there are up at the top here. 6 genes. That's not helpful. How many rows? There you go. 6, 12, 12 plus, no, 5, 11, 16. Lots of that. It looks similar. I can't do the count. I'm not going to do the count while we're sat here. But there isn't actually really any changes. So that's using R to do an equivalent lookup to what we can do on the website. What you wouldn't really do on the website is go and paste in 15,000 identifiers and do this kind of lookup. And that's what we're going to do now. Is we're going to now we're going to use BioMart to get a set of annotation for all of the genes that we've analysed for differential expression. So for all of our 15,000 genes in filter DF1. So to do that, it's just a case of constructing another one of these getBM calls in the same way as we have done before. So this time we're going to save it to a variable, which we're going to call annotation. We're going to run getBM. Okay, our attributes that we want back. So I don't want the transcript IDs back at this point. What I want is I want to get back one row for every gene in my data set. So I don't want to plug in any attributes here, which are going to give me more than one result per gene. So with that in mind, I'm going to get the ensemble gene ID, because we need to be able to map between our two tables. So we need the ensemble gene ID back. What else should we get? I could look this up in the list attributes table, but I know what I want, so I'm going to do this slightly quicker, especially as time is marching on. So ensemble gene ID, chromosome name. So let's get which chromosome the gene is on. Let's get the position. So let's get the start position. And let's get the end position. So that we know where on that chromosome the gene is. Let's get a couple of other things. Strand, whether it's positive or negative, on the positive strand or the negative strand. Let's get gene biotype. So that's whether it's a protein coding gene or whether it's a non-coding gene. There's lots of different biotypes for genes. Protein coding is the one that we're usually most interested in a gene expression experiment. Let's get the external gene name, because that's our human readable gene name, the thing that most people understand for genes. And let's also get the description to get a slightly longer description of the gene, which gives some hint to its function. So those are the attributes that we're going to recover. And every gene should only have one entry for each of these attributes. So each gene should only have one gene symbol, should only have one gene name, should only have a single description, should only be present in one chromosome at one location. So what we shouldn't end up with is multiple entries per gene as a result of this query. Okay, so there are our attributes. The next thing that we want to define are our filters. And our filters are the same as they were before. We're just going to filter on the ensemble gene ID, because those are the identifiers that we're passing in to our BioMart query. So what we're passing in as values, which is our next argument, is a list of ensemble gene IDs, which we get from our data frame. So filter df1 dollars ends gene. That's all of the ensemble gene IDs that we analysed for differential expression, the 15,000 genes that ended up on our volcano plot yesterday. So those are our values. And then the mart again is the same. So the mart is the our connection to BioMart that we made earlier on. So ensemble 99 is the variable that holds the information for that connection. Okay, so that is our getBM call to retrieve the annotation. How many we've got? 15,981 genes. We should end up with, hopefully, I'm going to keep my fingers crossed, I really hope this is going to be the case, we should end up with a data frame with 15,981 rows in it at the end of this query. This will take a minute or two because this is a big query to pass to BioMart. We're asking BioMart here for a lot of information. And so we get a little progress bar in our console, and it didn't take that long. So that has retrieved that annotation. So we now have a new variable, this annotation variable. Let's find out how many rows it's got. So n row annotation. So if this has worked as I expected, this should have, and it does, yay, 15,981 rows. 1 row per gene ID. And we can have a look at it. So we can use view to have a look at it, same as we have done other big data frames. There we go. And we can see, so ensemble gene ID 3 is on chromosome X at these positions. It's on the negative strand, so -1. It's a protein coding gene. It's T-span 6, which is tetraspanin 6, and that's all of that data that we retrieved for that first gene. Okay, and we have all of that information for all 16,000 genes that we looked up. Okay, I can see a couple of queries in the comments. If you're only using one filter query, ensemble gene ID in this case, is it necessary to use C? No, it's not necessary. I'm doing it for completeness, but no, you can leave the C off if it's just one thing. It's just if you had more than one filter, you would need to do it as a vector. But because we only have one, we don't need to do it as a vector. How do you know that ensemble needs quotes? So I think that was back a little bit. So here, ensemble needs quotes, but the version 99 doesn't. So for that, the answer to that is in the documentation for use ensemble, where it tells you, it should tell you, ensemble version to connect. It should really tell you that this should be a double rather than a string. But it doesn't. And there's not even an example. So the answer to that is I just knew. It may even cast a string to a double in that case, but I don't know for sure. The answer to that is I knew that because I've done it lots of times before. Sorry. Sometimes nothing quite trumps experience. Okay, so that's our annotation. And that's nice, but at the moment, it's completely disconnected from our results. In order to make use of it, we want to be able to connect it up to our results object. We can do that by merging these two data frames. They've got the same number of rows, and actually those rows are in the same order, but we shouldn't really trust that. We shouldn't really rely on that. But we can do a merge, and we can do it using the join methods that dplyr gives us access to. So dplyr is a package, it's a tidyverse package for working with tibbles and data frames that has methods for filtering data and for selecting out columns and slicing out rows, and we've used it a little bit in previous lessons. One of the things that it gives us is these methods called joins. And a join is terminology taken from databases. So from things like structured query languages give you access to joins, SQL. So we can use joins to combine two data frames, and we're going to use what's called a left join. If you want to know the difference between left joins, right joins, full joins, inner joins and outer joins and things like that, then I suggest you Google it, because I can't. So if we want our annotated data frame, so we're going to call it annotated DF. It's going to be a combination of a join between our filter df1. So we're going to left join filter df1 and our annotation. Now, if so, what the left join does is it adds the data on the right to the data on the left. So it adds the data in your second argument to the data in your first argument. If you did a right join, it would do it the other way around. Okay, it would add the data in your left to the data on the right. But we're doing a left join, so we're adding the annotation onto our filtered data frame. If these two data frames had a column name in common, then this would just work. So if they both had a column called ens gene, or if they both had a column called ensemble gene ID, then this would just work, and they would join up on the column that they have in common. But our two data frames don't have a column name in common, because filter df1, let's just view them both. Filter df1 has an ens gene column, which contains the ensemble gene IDs, and annotation has an ensemble gene ID column, which contains the ensemble gene IDs. They contain the equivalent data, but they're called different things. So we have to tell our join method that they're called different things. And the argument for doing that is the by argument. Okay? And the way that this works is really weird. It looks really weird to me as an experienced user of R. It may not look weird to you. What it takes is a vector, and it's a named vector where the names are the column names in the left dataset, and the elements of the vector are the column names in the right dataset. So we want to join our ens gene column to our ensemble gene ID column, and that's what that looks like. And it looks, to me, that looks really weird. To you, as a relatively new R user, maybe that looks a little bit less weird than it does to me. But that's us. This is us telling this left join method that these two columns, this one in filter df1 and this one in annotation, are equivalent columns. That they contain effectively the same data. So if the thing in ens gene matches the thing in ensemble gene ID, I want to combine those two rows into one single row in my resulting data frame. So this will produce me, again, a data frame this time called annotated DF, which has this annotated DF, which has 15,981 rows, just like the two data frames that have been combined to produce it. But now we've got 15 rows. Whereas if we look at filter df1 and annotation, see, they have 8 rows and 8 rows respectively. So we've combined those two 8 columns and 8 columns respectively. We've combined those two 8-column data frames into one single 15-column data frame. And that's because they share one in common. So they've each got one that's the same, and then they've got 7 that are different. So your final data frame contains the one that's in common, and then the 7 from filter df1 and the 7 from annotation, to give you 15 total columns. And so we can look at this annotated DF, and we can see now we have, so here is the entry for ensemble gene ID 0003. We've got the results for it from DSeq, so the base mean, the log2 fold change, the things which we've looked at previously. But then if we keep scrolling over to and we have the description of the gene included on the same row as our DSeq results. Okay, so we've retrieved the annotation and we've combined that annotation with our results in a way that means that it all makes sense. All of our rows in our data frame now make sense. And now we can use that information back in our plot that we drew yesterday. So let's redraw that plot again. So I'm just going to copy and paste this text here. Paste it in down at the bottom from yesterday. But instead of giving ggplot the ends gene ID as an extra aesthetic, we're going to give it the external gene name as an aesthetic. And what that means now, so for the ggplot, it doesn't mean anything. If we plot out the ggplot version, hold on, I gave it the wrong data frame. Sorry, I have to give it annotated df. Give it the right data frame, Simon. So we have to pass it the data frame which has the external gene name in it, otherwise it's not going to be able to find it. For the ggplot version of the plot, it makes no difference. It will plot it out and it will look exactly the same as it looked yesterday with no changes whatsoever. But the ggplotly version of the same plot, when we produce that, when we wait for my computer to think a little bit about what it's doing, consider its life, there we go, and we hover over, you will now find that in the tooltip, we have no longer do we have the ensemble gene ID, but we have the gene name that's human readable, and that tells us more specifically what that gene is. And I will leave that as today's coup de grace. So with that, I'm going to end today because it's already after 5 o'clock. I've had fun today. I've really enjoyed that. A bit seat in my pants. I've come off the back of 7 meetings today because my working from home plan of trying to reduce the number of meetings that I have really caught up with me today in a big way. So hopefully my voice has just about held out, and hopefully you have enjoyed today as much as I have. So that's it, and I will see you tomorrow. Tomorrow, I think maybe we might produce a heat map. I think that sounds like a fun thing to do. Okay, so see you later, everybody. Stay safe, keep well, and see you next time. Bye-bye.